Okay, so as we begin today, I'd like us to start by drawing a sarcomere. So I'd like you to pull out a blank section of your notebook. Go ahead and draw one sarcomere and label each of these pieces. Okay, so for our review of the sarcomere, I'm going to go ahead and draw along with you. The reason we want to make sure that we know how to draw a sarcomere is because when we can draw a sarcomere, this is going to emphasize to us how that contraction happens, right? And we're going to be able to understand which of these zones would stay the same size as our sarcomeres contract and which of them are going to become smaller as we have this sliding filament model where we have our cross bridge cycling pulling together the actin at the ends of our sarcomere and pulling everything closer. So our sarcomere is measured from Z line to Z line. So we got our jagged lines, our zigzags. So this is one Z line. And then we'll have another Z line on the other end. Okay. Okay. Our M line is going to be in the center. Okay. And I like to do them in this order because this reminds me where the filaments are attached to each other. So the Z line, we have our actin or thin filaments. And the M line, we have our myosin, our thick filaments. So next, we're going to draw on the filaments themselves. Okay. So we are going to add in our thin filament, the actin, projecting. Okay. All right. So we got our actin, thin filament here. Okay. Extending towards the center of our sarcomere from that Z line. That's one of our two contractile proteins. And then our other is going to be in the center, spanning across. And here we have that myosin, that thick filament. Okay, spanning across. And we might also draw on our cross bridges. That's going to help us understand how we pull this sarcomere shorter. I'm drawing on those cross bridges, those heads of the myosin. Got something like that. Something like that. Okay. That's going to lead us to our bands. So our bands, we're thinking about where we have actin, where we have myosin, and where we have our regions of overlap. So our A band is anywhere we have our thick filament, so anywhere we have our myosin. So actually, let's go ahead and label this versus this. Okay. So in orange here, we have our thick filament, which is myosin. Whoops. It does say myosin. Here we have our thin filament, our actin. So for the A band, we are going to select everywhere there is myosin. We don't care whether we have actin or not. All right, so our A band would extend just the full length of the myosin. Okay, just the length of the myosin. This is our A band. So, question for y'all, is this going to shorten as our sarcomere contracts, this A band? No, this doesn't shorten because the myosin doesn't actually change its length. Right? The myosin itself stays the same size. The reason our sarcomere is shortening is because those Z lines, that actin, is getting pulled and slid towards the center. Right? So that doesn't affect the A band because we're not paying attention to are they overlapping or not. The A band is just how long is this molecule, essentially. So that's our A band. Okay. So our H zone, we do care about whether we have overlap. So the H zone is only where we have myosin, but no actin. Okay. Myosin, but no actin gives us the H zone. Now, does this change size as our sarcomere contracts? Answer is yes, right? So as we have actin, these blue lines move towards our M line, towards the center. We are going to be decreasing this amount of space where we have only myosin, but no actin, right? So this is going to decrease. We're going to be increasing the region of overlap. So conversely, our region of no overlap is shrinking. Okay, so the H zone would shrink as the sarcomere contracts. Finally, we have our I band. Our I band is where we have only thin filament, only actin, no overlap. Our I band would be here and would reach into the next sarcomere as well. So this is our I band. Okay, so yes, no, does this change size as we contract? Yes, right? We're again thinking about a region where there is no overlap, right? So as our cross-bridge cycle pulls the sarcomere shorter, slides together our filaments, this region where we have only actin and no myosin is going to shrink. And now that we know about the cross-bridge cycle and the sliding filament model, we can hopefully understand a little bit more why we care about these zones, why we might bother naming them. So we had a slide in last week's lecture that looked like this. So this is just a better drawing of what's happening as we have our sarcomere shortening. So we can see there how those regions of overlap are changing, right? So that H zone where we had only myosin got much shorter, right? That region of no overlap. I band also got shorter, right? I band got shorter a bit. Okay. But the H zone, or sorry, the A band stays the same size because that's dependent only on the length of the myosin and has nothing to do with the overlap. So last time we talked about the cross-bridge cycle, and probably on Wednesday, we will do an activity to review that. But before we do, I want to think a little bit about how we turn that cycle on, okay? So we're going to think a little bit about why our muscle is contracting and then pair it up with that cycle, that how to let a muscle contract. Okay. So this is called excitation contraction coupling. This is how we turn a muscle contraction on, essentially. So we have a sequence of events, just like we've had at our synapses, like we've had at our neuromuscular junction, right? We're going to have steps to turn on a muscle, tell these sarcomeres to get shorter. Okay. So this signal, excitation part, right, is coming down a somatic efferent nerve, right? So somatic because we're going to skeletal muscle, so that voluntary type of muscle, that type that you think about and contract it. Okay. So we're coming down that somatic nerve, efferent because it's going away from the central nervous system. It's going to go to that neuromuscular junction with our muscle. Right, so we have our muscle side as well. And on the muscle side is where we will have the contraction part. So the contraction part is that we're trying to turn on the cross-bridge cycle. Okay. So we're dependent on the neural input. We need an action potential to come down that somatic nerve. What we're going to do in our muscle cell, in our muscle fiber, is we're going to be releasing calcium from the sarcoplasmic reticulum. And that calcium, so it lets us push the tropomyosin aside, that long sort of pink spaghetti strand that was lying over our myosin binding sites on the actin. We need the calcium to push that aside. And that is what allows us to bind actin and myosin together to begin our cross-bridge cycle. Okay. So for our steps, what we're going to be doing, right? We talked about the neuromuscular junction. So those steps would happen first. We'd have that action potential coming down our somatic nerve. We'd have the acetylcholine being released from that somatic nerve. And then we have our nicotinic cholinergic receptor on our motor end plate on the muscle side, receiving that acetylcholine and reacting. So this next part is all a reaction in the muscle cell. So we've received that signal in the form of acetylcholine on our muscle side. So now we are going to have an action potential in the muscle cell itself. So this is turning on an action potential in our muscle cell. And the sarcolemma, right, was one of our terms from last time. So this is the word basically for our plasma membrane now in a muscle cell specifically. So we're going to have an action potential traveling down the border of the sarcolemma, so down the border of our muscle fiber cell membrane. Okay. So that's happening first. Then that action potential is going to travel into our muscle fiber. So into the muscle fiber, down a T tubule. So down in our picture, they were green before. So down one of these structures that tunnels into the muscle fiber. Between our sarcoplasmic reticulum, sort of, we saw those triads where we had a T tubule in the center of two lateral tracts of the sarcoplasmic reticulum. Okay. Then we're going to have a receptor in the T tubule that opens calcium channels. Okay. So opening calcium channels in the lateral sacs of the sarcoplasmic reticulum. Then that calcium is going to be released, so it's going to be increasing in the cytosol, so it's increasing in the inside of our muscle cell. When we have calcium floating around in the cytoplasm and the cytosol, it's now going to bump in to the troponin on our actin filaments. As it bumps into that troponin, binds to it, shifts tropomyosin aside, exposing the myosin binding sites, which allows us to begin the cross-bridge cycle. So we'll take a look at this now. So here's what this whole cycle looks like. So we can see that our goal, right, is to turn on this cross-bridge cycle. So this is showing us that cross-bridge cycle. And we can see this neuromuscular junction up at the top. So we can see that somatic nerve coming in. And here we see a zoomed in vision of some of these structures in the muscle fiber. We're going to go through one by one. Okay. So the first thing that's happening is that action of the somatic nerve at the neuromuscular junction. Okay. So we had our axon terminal, which we also called a terminal bouton, right, from our somatic nerve. So that terminal bouton, axon terminal of the neuromuscular junction. Goal is to release our neurotransmitter, which is acetylcholine, right? So we're releasing that acetylcholine, and it's binding to receptors here on the motor end plate. The motor end plate is the part of that muscle fiber that has the receptors that are able to receive that acetylcholine. When we receive that acetylcholine, when we bind the acetylcholine on the muscle cell side, what we do is we create an end plate potential. So motor end plate, so end plate potential, right? End plate potential, basically kicking off an action potential in our muscle cell. So this is an excitatory connection here between our somatic nerve and our muscle fiber. So we are creating an action potential. We're depolarizing this muscle fiber. That's excitation. So these are excitatory. So next, we have this action potential. So basically this wave of charge, right? Traveling along the membrane. So the sarcolemma is our membrane. So we're traveling down it, just like we had action potentials propagate down an axon in our neurons. Right, so this action potential is propagating down the sarcolemma, and it's going into our T-tubules. So this is the T-tubule, right? So that action potential propagating down the T-tubule, and then the T-tubule dips through our sarcoplasmic reticulum, which is the stuff in purple. Okay. So the T-tubule is in a triad with the sarcoplasmic reticulum. So as this action potential travels, we're going to be able to affect the sarcoplasmic reticulum. Okay. So our goal here in carrying this action potential down T-tubule through the sarcoplasmic reticulum is to release calcium. So that's what we have, these little green dots here. We're releasing calcium. Okay. We're releasing calcium. And I don't actually care if you know the receptor names in the T-tubule and the sarcoplasmic reticulum. What I do want you to know is this part about the calcium release. So that's our goal. This action potential coming down the T-tubule is ultimately triggering our calcium release from the sarcoplasmic reticulum. So calcium has been stored here in this structure. So stored in the sarcoplasmic reticulum, now we're moving it into the cytosol. Okay. So once we have calcium in that cytosol, we're going to bind the calcium onto troponin. So troponin is this little snowman guy, troponin. Okay, so we can see calcium bound to the top of it now. Okay, when calcium binds to troponin, this is like a button pushing to the side, or maybe like a switch, right? So as it shifts because of the binding of the calcium, you can think calcium is like your finger on a switch pushing it to the side. Right, so that's what the calcium is doing. So then that troponin pushes aside the tropomyosin. The tropomyosin was this long strand, right, which is in pink. Previously, it was lying over top of our myosin binding site, so over top of these dark spots. But when the calcium is bound, it's pushed to the side. So these myosin binding sites are now exposed. So we've exposed these myosin binding sites. Okay. So now that we have exposed these myosin binding sites, we can begin our cross-bridge cycle. We're beginning our cross-bridge cycle. This is where we have those cross bridges, those heads on the myosin. They're actin binding sites are binding to the myosin binding sites on the actin. Ultimately, we're pushing aside the actin, so pushing it towards that center line, pushing it towards the M line, pushing it towards the center of the sarcomere. Right, so what we have here is a picture of the power stroke. We have a picture of that push, right? So cross-bridge cycle pushes, and then as the second part of the cross-bridge cycle, we take in an ATP molecule, bind it to our ATPase site and break it apart for energy. So we go into our high energy state for our myosin, ready to do this again. Okay. So all of this is triggered by that calcium coming out of the sarcoplasmic reticulum, binding to the troponin, exposing those binding sites, making all of this possible. All right. And then eventually we have that calcium transported back into the sarcoplasmic reticulum. So we're reharvesting that calcium and saving it for later. And so as our action potential diminishes, right, as it fades away, we take this calcium and we shovel it back into the sarcoplasmic reticulum. So we're waiting for our next action potential to essentially tell us to do this cross-bridge cycle stuff again. Right? So as the calcium in the cytosol fades away, your muscle is relaxing because we're going to get rid of the calcium bound to the troponin. covering back over the myosin binding sites, meaning we can't connect to the actin anymore, meaning our sarcomere will lengthen again. Okay. So here we have that tropomyosin as we remove the calcium going back over the binding site. So you can see the difference in that state here. All right, when the calcium was bound, we could see our binding sites, our myosin binding sites on actin, but now we can see they're all crossed out, right? They're crossed over by that tropomyosin because we don't have any calcium bound anymore, right? Before we had a calcium here on our troponin, now we do not, right? So that's the whole process of how we take an action potential coming down our somatic efferent nerve, joining into our neuromuscular junction, ultimately releasing calcium inside the muscle fiber in order to start our cross-bridge cycle, and then letting the muscle relax as we suck calcium back into our sarcoplasmic reticulum. So we can zoom in a bit on those states of the actin, because I know they were real small in that big cycle picture. Okay, so what we were looking at was the relationship between our troponin complex, right? So our troponin complex, oops, pick a different color, our tropomyosin, right? Our tropomyosin, right? That's this part, and our actin, but specifically our myosin binding sites we were interested in. Right? The myosin binding sites are these black pieces. So in the relaxed state, we have tropomyosin covering over our myosin binding sites. So we have no ability to do the cross-bridge cycle because the binding site is covered, so it couldn't bind onto myosin if it tried. Okay? Covered over, just can't do it. When we have calcium, though, we have a shift. So the troponin complex has shifted as it's bound to calcium. And that shift has linked it up with the tropomyosin, right? So this is the tropomyosin. Tropomyosin, okay? So we can now see the exposure of those myosin binding sites on the actin, right? So that is the point of excitation contraction coupling. We're coupling the excitation to this action potential, creating a change in membrane potential, right, to the physical contraction of the muscle, which is the cross-bridge cycle. So we're using this action potential to trigger the cross-bridge cycle. So in order to terminate our contraction, so in order to relax the muscle, we're going to have to get rid of the calcium. Okay? So when we get rid of the calcium, this is going to put the tropomyosin back into place. So it's going to cross out those myosin binding sites again, which are on the actin. Okay. So we do have some things that help us remove that calcium from the cytosol. One of them is in the sarcoplasmic reticulum. So we have a calcium ATPase sucking back up the calcium back into that sarcoplasmic reticulum. Let's take a quick look here. Okay. We have to watch an ad. So we'll look at this in three-ish D.
People trying to explain the events of the cross-bridge cycle. Each of the series of events responsible for the actual contraction of the muscle. Here we see a muscle. And one single muscle itself. A closer look at the muscle fiber shows that this one single muscle we see one line. Looking closer at the muscle we'll focus on let me point out the Z is. If here is they have lines in the two-dimensional, but seeing the Z is the boundary sarcomere is the structure of the actual basic. In this image, we see most of the structure is one structure. We have here and here, Z here, marking the boundaries of the sarcomere. In this closer view, we see two distinct structures. The thick film and mice and one block shade. We also see the thin film. These two films actually form. Before we look at the media, how these structures work together, let's zoom in. Read the notice how mice and head move the element. Then move the thin filament toward the center of the sarcomere. If you watch the sequence this time, you only get a cover of dead shot for many years. To begin the crossroad cycle, we need to find an answer to the structure. This arises in the form of nerve stimulation, which leads to the release of calcium and internal circulation. The increase in calcium concentration is not part of sprint cycle proceeding. Does calcium get the cross-bridge cycle started? Well, at rest, when the calcium concentration bridges cannot form. This is because tropomyosin is what is seen here. The active site. These active sites are where myosin gas would bind to form cross bridges between the state and the development. When calcium concentration is high, the active site is exposed. This occurs through two intermediate steps. First, calcium binds protein following protein change in shape. Second, the shape change in protein alters the position of tropomyosin. In this new position, tropomyosin no longer covers the active sites. Now that the active sites are exposed, the cross-bridge cycle begins. We'll go through one complete cross-bridge cycle, which consists of four distinct steps. The first step, seen on this slide, is cross-bridge formation. The myosin head binds the actin, forming the cross-bridge between the patient and the woman. Besides myosin bound actin, we also see an ADP and an organic phosphate attached to the myosin. These were left by a previous cross-bridge cycle. In step two, the myosin head pivots using energy that was previously stored. This movement is called the power stroke. The power stroke pulls the actin thin filament toward the center of the sarcomere, shortening the overall distance. Power stroke occurs, ADP is released, freeing up space right about here for ATP to bind. We begin step three as ATP binds. This triggers detachment or breaking of the cross bridge. This is the first of the two major roles for ATP cross bridge detachment. Note that the binding of ATP to myosin is sufficient to break the cross bridge. The ATP itself is unchanged so far. Not until the last step of the cycle is bound ATP hydrolyzed to ADP and inorganic phosphate. The energy released from this hydrolysis will be used to re-top the myosin head, storing energy to be used in the power stroke of the next cross-bridge cycle. At this point, we've completed one cross-bridge cycle. The calcium is still present, as it is right here. The active site are still exposed, and a new cross-bridge cycle will form. The cycle will continue. Now, you may wonder, myosin gets detached. It is right here. Why the thin filaments don't simply slide back? Remember, we've been focusing on thousands of any single myofiber. They don't release. Some cross-bridges will release while others still attached. Many cross-bridges work together to shorten each sarcomere and ultimately the whole muscle. The cross-bridge cycle will continue as long as two factors are present. The first is an elevated concentration of calcium in and around the myofibril. Recall that this rise in calcium level is a consequence of nerve input. Second, adequate supply of ATP must be present. ATP is directly responsible for attachment and ultimately ATP hydrolysis powers sarcomere shortening.
So don't get hung up on the number of steps, so long as you understand this process and what happens first, what happens next, you should be good to go. As you can see in a lot of these processes, people break them up into differing levels of detail. Don't think that link went exactly where I expected it to, but you got to see the myosin kind of walking, which is the fun part, right? Okay. So we're going to discuss briefly the overall mechanism of skeletal contraction now. Okay. So when you have this contraction triggered, right, have this nervous impulse coming in, we're going to have a contraction in a muscle fiber in response to a single action potential coming down our motor nerve. Okay. So this is going to elicit something called the twitch, right? So like when your eye twitches, right? Like it's contracting over and over and over again, right? This is the twitch. Your muscle is twitching. Okay. So this is going to be an all or nothing event for the muscle fiber at rest, which is to say it's either going to happen or it's not going to happen. Okay. We can mention it for a muscle fiber as a whole. So we could say a muscle fiber is twitching, or we could be more specific, say a specific motor unit is twitching. All right, so a motor unit, remember, is our different muscle cells that all go back to a single motor nerve, but we actually have many motor nerves coming to a named muscle, right? So we could also have an entire named muscle having a twitch. So we have phases for our twitch as well. So at the beginning, we have our latent period. So this refers to the fact that it takes a little bit of time from the time our action potential is coming down our motor nerve, linking up with our muscle cell, helping us release calcium into the cytosol, to when we actually elicit contraction. So when our muscle actually starts to shorten as our actin is sliding past the myosin and shortening our sarcomeres. So this takes about a few milliseconds before this starts happening, right? So during the latent period, we're doing our whole process of excitation, contraction, coupling. We're trying to kickstart those cross-bridge cycles. Then we have our actual contraction phase. So this is the time period where our cross-bridge cycle is going. So it's going around a number of times, lots of times. We have lots of cross bridges forming at all our various myosin heads. Okay. So we're increasing what we refer to as tension. Right? So we're increasing like this pull at the ends of the muscle. Okay. This is about 10 to 100 milliseconds. Then your muscle relaxes. It relaxes because that calcium is letting go of our troponin, which means our tropomyosin is covering over those myosin binding sites again. Right, so as we do this, our sarcomere goes back into its sort of like optimal length, or its resting length, right? So your muscle is contracting or relaxing. And there's going to be a decrease in tension, right? This takes more time than the contraction phase, but we're not thinking about any specific length of time. So if we were to graph it, what we might graph is something we can actually measure, which is tension, right? So you could actually measure force of this muscle essentially. Right? So the force of this muscle contraction, you could attach like a weight or some kind of transponder, right? See how much tension, how much force is being produced. So you'd look at this over time if we're trying to look at the phases of the twitch. So we have time over here on the x-axis. So we have tension, which is a measure of the force of contraction, over on the y-axis. So first we'd have our stimulus. So essentially our thought telling our motor nerve, telling us to contract, right? So we're sending that action potential down our motor nerve. Okay. We're doing our excitation contraction coupling during this latent period. So we're not going to see any increases in tension here because we're waiting for that nervous impulse to release calcium to start the cross-bridge cycle. During the contraction phase, we see more and more cross bridges forming, right? Pulling on the actin more and more. So that's our contraction phase. And during this time, we see our tension increasing, right? So forming as many cross bridges as we can. And as we form more, we pull the actin further and further towards that M line, contracting the muscle, making that sarcomere shorter and shorter. As calcium starts to unbind, we start to relax. Takes us a bit longer, relaxation phase, as we're sucking up that calcium back into our sarcoplasmic reticulum, covering over those myosin binding sites with our tropomyosin. So when we talk about twitches, we will sometimes distinguish something called an isometric twitch from an isotonic twitch. So when we think about isometric and isotonic twitches, what we're really going to be thinking about, like, is something moving or are you holding something in place? Kind of. We'll go through each, and so I'll explain what I mean. So we want to remember that we have our contractile element, so the stuff that's actually shortening our sarcomeres. Okay. But when we think about a muscle as a whole, we actually have some other things going on, right? So in lab, we've seen some muscles, right? So we have our muscle, right? I'm going to attach it to a bone on the other end. Okay. My bone will be yellow. I'm going to draw a really bad femur. Okay. Cool. Okay. So we're attaching to a bone on one end. Just draw that attachment there. We're going to attach to another bone on the other end. But before we do that, instead of having just our muscle belly, which is where we have our sarcomeres and our muscle fibers, right? So the muscle belly is going to have the sarcomeres, those fascicles, all those different pieces. We're also going to have some connective tissue and tendon at the other end, right? So when you looked in lab, you saw sort of shiny white lines at the ends of the muscle belly. So we looked at those tendons like going into the fingers, right? When you saw the, for example, the extensor digitorum splitting out into its many tendons, right? That shiny white part is part of our named muscle, but it's not full of sarcomeres. It's a different type of tissue. So it's some type of connective tissue. So specifically, we call those ones that type of connective tissue a tendon, right? So that tendon we could refer to as an elastic element, okay? So they're in series, which is to say that they're next to each other, right? So we have a muscle belly, and then we have that tendon in series, so next to each other. So the place that we're going to be creating the force is with the contraction of the motor fibers in this muscle belly. But we do also have this tendon, which is elastic, and it can kind of lengthen a bit, right? So they are tough, right? Like you did feel that they are tough, but they do have some elasticity there. They can change their length. So we can have times, all right, like setting my hand on this desk, right? I'm contracting muscle and I'm pushing, but nothing's actually moving, right? So this is because I'm contracting my muscle fibers, but we don't have movement because we're braced at one end, meaning that what's actually changing is the length of the elastic part, the tendon, rather than actually moving something like through the air. So the force exerted by the contracting muscle is the tension coming from those sarcomeres in the muscle belly. But when we're thinking about movement of a load or of an object, we have to remember that the tendon exists too. Okay, so a load, I should say, I'll draw, instead of another bone, I'm going to draw like a really bad little dumbbell thing. All right, so the load is at the opposite end, right? I load over here, okay? So we're thinking about loads and contraction and tension as we think about the twitch and as we differentiate our isometric from our isotonic twitches. So we're going to start with the isometric twitch. So if you do like an isometric exercise, right, that's one where you're like maybe not moving. So think about like planking, right? It's hard, but you don't look like you're doing anything, okay? So an isometric twitch is a contraction where our length overall of that like named muscle, including its tendon, is staying the same length. So we're going to have the length remaining constant, but the contractile element, the sarcomeres, are still contracted. So we're still generating tension, right? We're still generating some type of force, but we are not moving our load. So we might express this mathematically as saying like our load is greater than our tension. Okay, so your load here might be in terms of like a physical exercise, your load might be like your body weight or the ground or something like that. It might be something you can't move. Okay. But your load is greater than the tension of the muscle, isometric. Okay. So we're going to stretch our elastic elements with the isometric twitch. So we're like stretching tendons, but the overall muscle is not shortening, which means that the load is not getting lifted. Okay. So we're just going to look at a picture and we'll start with isotonic next time. Okay, but we can see what would happen there just as an example of what I mean, right? Like here we have the muscle belly. We have an isometric twitch. The belly of this muscle got shorter, but the tendon got longer, right? The tendon got longer. So these force plates at either end like didn't actually move because the overall length stayed the same. So I'll pick up here.